All right. This is so nice. Thank you so much for having me and giving me this opportunity. So, keeping in lines with the theme of the symposium, I want to share some of our work on human monoclonals to SARS-CoV-2. So, when we started the work, we had two things in mind. One was that human monoclonals, obviously uncontested, are very good therapeutics, and they fill the gap that vaccines cannot reach. One, like immunocompromised individuals, or the aged or very young that do not mount a good immune response, or even the individuals that are vaccine hesitant. But the second point that we were really interested in is that they are highly expensive. So even for the developed world, it's a very expensive therapy to have. And for low and middle income countries, even though efforts are being made to make them more affordable, currently we are not there yet. So one straightforward thing we thought was to make them indigenously in places where they are needed, like in India. So that's where we started. So let me quickly show you where we are going, and what we've done this so far. So nobody needs to know this. We all know what antibodies do, but essentially they do a lot of cellular functions. But what we are primarily interested, at least in this context, is neutralization. And neutralization, as we all are aware, is essentially making the virus ineffective. Antibodies can do this in primarily two different ways. One could be just a game of numbers, where tons of antibodies will go and bind to a virus, or it could be in case of certain antibodies will bind to key epitopes on the virus and not let it reduce its infectivity. So in case of SARS-CoV-2, this is the spike protein. Again, all of us know this. Antibodies can target different regions of the spike protein, three of which that we are very well aware of. One is the S2 subunit, one is the N-terminal domain, where people have found antibody responses, and the third is the receptor binding domain, or the RBD. This is the domain, as we all know, is the one that interacts with the host ACE2 receptor and the virus infects the cell. So this is the RBD domain that we were interested in mining for human monoclonal antibodies, considering that we would probably find most of the neutralizing antibodies here and probably find something that could be of therapeutic value. So when we started, just like everyone else, we didn't know what was the SARS-CoV-2 response like. So one of the first questions we asked was, what does the RBD specific antibody response look like in COVID-19 recovered individuals? And the reason we decided to go with recovered individuals is that we knew that SARS-CoV-2 is a primary infection. And therefore, to find anything that was of therapeutic value, probably we should be looking at the memory response. So we were interested in baiting for memory B cells. So we started with recovered individuals, where that was somewhere between 6 to 8 weeks post-recovery. At that point, it was a guess that we should have some level of memory responses by then. And so quickly to go through this slide, when we recruited individuals, we essentially saw a heterogeneity in their IgG titers. What I'm showing you here is basically RBD specific IgG titers. This is essentially an in-house ELISA. RBD has been quoted at about 1 microgram per ml. And then we've calculated the minimum binding dilution in the plasma and compared it to pre-pandemic healthy. And as you can see, some people have no responses and some people had very high responses. What was interesting is that when we compared these IgG responses on the y-axis to neutralizing antibody titers to SARS-CoV-2 live virus, we noticed that only when you reach a titer of about 3,000 IgG titers do you start seeing some sort of a correlation with neutralizing antibody titers. And this was the take-home message we had in this paper that we published recently. So these were the individuals here that we then picked to then make human monoclonal antibodies by baiting their memory B cells. We chose 5 individuals from here, and that was based not only on their IgG and their neutralizing antibodies, but some logistical things like where we could recover more PBMCs, which had live PBMCs, and the staining looked clean so that we could do single cell sorts. So here are the 5 individuals right here, subject 1, 2, 3, 4, 5, and I'm comparing them to healthy pre-pandemic individuals. The first row that I'm showing you here is basically their G, M, and A titers. And the numericals in the box is basically their titers for IgG. This is their neutralizing antibody titers. Again, this is the IC50 value in the box. And these were the memory B cells that we baited. Each one of them, the frequency of memory B cells is written within the plot right here. And the staining was done by directly conjugating the mammalian expressed RBD to FITC, and then we did a direct stain. So this is the gating strategy for making the human monoclonals. So we essentially, there are multiple ways that people can make human monoclonals. This is the way we follow in the lab, where we do single cell sorts of the B cells of interest. In this case, as I mentioned, it is the memory B cells. So we take all total CD19 positive cells that are CD3 negative. And since we are interested in memory and they'll be class switched, we go for the IgD negative cells. And then we may or may not do other stains like CD27, etc. But in this case, we straight went for staining for RBD and gated on these cells. And then you do a single cell sort in a 96 well plate. The plate contains capture buffer. So as soon as your cell falls into it, it will lyse, RNA is released, and we go ahead and do the cDNA synthesis on the same day. So it's a kind of a long day. And this is the protocol we follow. Most of you would be aware of this protocol. After cDNA synthesis, the entire thing goes through multiple rounds of sequencing and cloning. And then we clone them into 2 different expression vectors. These are mammalian expression vectors, one for the heavy chain, one for the light chain. Then we bring it back in a transfection. These are either XP293 cells or X293F cells. The antibody is found in the supernatant, which we purify, and that's what it looks like. So every monoclonal that we make, we can go back to the B cell that we had sorted in the well, in the 96 well plate. So we did this for the 5 subjects and found 204 productive clones, out of which 92 were RBD binders. And so what I'm showing you here essentially on the x-axis is monoclonal concentration starting from 1 microgram per ml. And their OD on the y-axis. And this again is the ELISA is coated with 1 microgram of RBD. As you can appreciate, we did find some very nice strong binders and some of them were weak. If we calculate the minimum binding concentration of these monoclonals, some of them are really nice binders and their concentration comes out to be about 0.001 microgram per ml. And these isotype controls are other dengue, chikungunya, and other monoclonals that we are making in the lab. We just picked few of them just to see what our background looked like. So since SARS-CoV-2 belongs to the family of coronaviruses, as we all know, the first thing we were interested in was that these monoclonals bind to SARS-CoV-2 RBD, but what else do they bind to? Do they bind to RBD of any other coronavirus? So that's what we did. So what I'm showing you here is essentially picked up from Mehul's paper that he had published. And they showed the RBD of different coronaviruses and what their sequence similarity is. I just thought I'll throw it in here just for reference. But as you can appreciate, from CoV-2 to CoV-1, there's already a dissimilarity of about 25%, and that keeps continuing to go down. So what we had the RBD for SARS-CoV-2, we cloned the RBD of CoV-1, MERS, and 2 circulating coronaviruses, 1 beta, OC43, and 1 NL63. And this is what we found. Again, RBD was coated at 1 microgram per ml. And then we tested all our 92 monoclonals at 1 concentration of 10 micrograms per ml. And all the monoclonals are sitting up here. These are the clone names with some controls. And this is our CoV-2 binding, which is in the top row. And when you look at CoV-1, you do see, based on the sequence similarity, some level of cross-reactivity with CoV-1. We didn't find any monoclonal that cross-reacted with MERS. But interestingly, even though there's barely any sequence similarity in the RBD, we found 1 monoclonal that is quite detectable in terms of its cross-reactivity to these 2 circulating coronaviruses, but the others were slightly milder. We are interested in seeing where exactly does it bind to, that if you have only this much sequence similarity, where exactly would this clone go and bind to? And it would be just interesting to know in terms of the antigen and the virus interaction and the antibody interaction. So while we make monoclonals, because we go through a lot of sequencing steps, there is a lot of other sequence information about the antibodies that we get. And this essentially gives us a lot of readout of the B cell response itself that these individuals have. So another question that we were interested in understanding was what was the diversity of this B cell response and the immunoglobulin gene usage. This tells us also if it's a very public gene usage or is it all constricted to a particular set of antibodies that are being made. So before I show you the data, one quick introduction to what I'm talking about, just in case there are some students in the audience. As we all know, the human B cell receptor repertoire can be about 10 to the 11 to 10 to the 12. And we don't have so many genes in the human body to account for so many antibodies being made and so many unique antibodies being made. So how does the immune system cope with this or deal with this is essentially by this process. One of the main processes that we all would have learned in our immunology class is VDJ recombination. So just quickly, antibodies are made of two heavy chains and two light chains. And these light chains can be either kappa or lambda. They have a variable region and they have a constant region. This variable region is where the antigen binds. This is the Fab. And this variable region consists of different gene segments. You could have about 40 variable diversity, 27, another joining 6. And then in the light chain, depending on whether you're a kappa or lambda, you could have 30 to 40 variable segments and 4 to 5 joining segments. And permutation combinations of both these VDJ on this side and VJ on this side leads to an increase in diversity to about 10 to the 8. So this is what we call as combinatorial diversity. Now, while VDJ recombination is happening, at the junctions of VDJ, you can have some level of amino acids being replaced or deleted or added, and further junctional diversity comes in at that point, and you can get further increase in repertoire. The third way that you can have an increase in diversity is when a naive B cell encounters an antigen and starts to go through the germinal center reaction, you have something called somatic hypermutations. And these somatic hypermutations can happen at a rate of about 10 to the minus 3, which is about 1,000,000-fold more than the spontaneous rate of mutation. So because we have sequence information, we can look at what our VDJ looks like, what is the gene usage there, and we can also look at somatic hypermutations to get a flavor of what the diversity of the antibody response looks like. So with that long drawn introduction, let me just quickly show you the immunogenetic information of these 92 RBDs. These donut plots essentially are heavy chain here and lambda and kappa light chain here. These two numbers should add up to 92. And as you can see, we have a healthy distribution of VH gene usage, with VH3 being most dominant. Now, if we look at V versus J, this is what it looks like. And this is again heavy chain, lambda and kappa. And what we see is basically these, let me quickly just explain what these heat maps are. These heat maps are basically generated from the frequency with which we find these gene sequences in the total frequency. And for the ease of presentation, we transform them into Z-scores. So darker the color, that means that particular gene segment, we found it again and again in our 92 monoclonals. And this means something. For example, if you look at the 3-66 or 3-53, these are monoclonals that are known to be very potent. This gene usage comes up again and again in the potent monoclonals which have gone into clinical trial. Whereas monoclonals that have a gene usage of 3-30 or 1-69 have been shown to be more broadly cross-reactive. So as we get this kind of data, we can then go back and either look at the literature of SARS-CoV-2 monoclonals or other studies of HIV and influenza to better understand the B cell response to SARS-CoV-2. The second thing I mentioned was somatic hypermutations, where as affinity maturation happens, SHM continues to grow. So when we looked at somatic hypermutations, if we do standalone heavy and light chain, it was pretty low. Most of the monoclonals lay below 5%. And if we do the same thing based on light chain on the y-axis and heavy chain on the x-axis, you'll appreciate the same thing. Most of our 92 monoclonals have very low SHM frequency. Now, this is not unexpected because SARS-CoV-2, as we all know, is a primary infection. I just mentioned it in the talk earlier. And therefore, it's a naive repertoire that is expanding. It's not a memory repertoire unless and until there were some cross-reactive clones that were expanding. And so this is not unexpected. What we know from other infections is that when you have a primary response and a naive repertoire is expanding with low SHM frequency, it kind of loosely correlates to lower binding capability, slightly lower neutralizing capability. So now our next question was, do these 92 RBD specific human monoclonals, do they even neutralize? So that is what we asked. Are these antibodies neutralizing? So the way we first looked at it was by just doing a ACE2 competition. This is by the standard gene script kit that many of us would have used. It's a surrogate for neutralizing antibodies. And what we saw is that also all the monoclonals are on the x-axis and percent ACE2 inhibition is on the y-axis, essentially calculated the way the manufacturer has said that we should. This dotted line gives you the cutoff that the kit has provided, that anything above this would be neutralizing or at least can abrogate ACE2 RBD interaction. And the one thing you would appreciate from here is that more than 50% of our monoclonals did compete with the ACE2 RBD interaction. So potentially they would be neutralizing clones. So that gave us some amount of confidence in our monoclonals. So we sent these monoclonals to Emory University because this project was in collaboration with Emory. And they did the live neutralization assays for these 92 MABs. And the ones that I'm showing you here is because I want to focus on these further in the talk, is that many of them were neutralizing, but 4 of them were very potent. So the 4 monoclonals I'm showing you here, these are their graphs, and this is the IC50 cutoff. And these are the calculated IC50s of each one of these monoclonals with their IDs mentioned here. This color code I'll continue to use in the next few slides, but essentially we found 4 monoclonals with one at about 0.028 micrograms per ml. So they were highly potent. So we were very happy about this. So Mehul's lab at Emory University ran these assays, and the graph I'm showing you here is to the original wild type Wuhan strain virus. So neutralization can happen in multiple ways, but the 2 ways that, as I mentioned in one of the first slides, is where the antibody binds, and the second is with what affinity does it bind. And affinity makes a huge difference is because when the monoclonal will bind to a virus and when it's taken up by a cell, then you don't want it to dissociate at that point and facilitate viral infection. So we looked at affinity and also a little bit on where these monoclonals were binding. And we focused on these 4 potent monoclonals. So what I'll do is quickly show you first the affinity data and then some amount of where they bind. So this is a BLI octet experiment that I would like to share with you. This was done with the monoclonal at 5 micrograms per ml, which were immobilized on protein A sensors. And the monoclonals are mentioned in the same color code on the top in each one of the columns with their IC50 values as calculated in the graph. The top row is RBD, the bottom row is spike hexapro. And the binding kinetics, we studied them with 300 seconds of association followed by 600 seconds of dissociation. And as you can see, and I have boxed MAB 13 because you can appreciate that both for RBD and for spike hexapro, there was barely any dissociation that we found. This was extremely potent and the affinities were calculated at about 10 to the minus 12 as compared to this monoclonal where you start to see a dissociation with spike and even with RBD. So this was one of the monoclonals that we think is extremely promising just based on its affinity and also actually based on where it binds. So now let me share some amount of structures that we have done for the same. So this is the cryo-EM analysis. This was also done at Emory in Eric Auckland's lab. Anamika is one of a really good structural biologist who did this work. And what I'm showing you here is cryo-EM micrographs that were generated on a Talos Arctica machine, and the resolution is of about 4 angstroms. The 2D classifications were generated after particle alignment, averaging, and then initial model generation was done for the 3D classes. And these 3D classes, the class 2 and the class 4 that is shown here, Anamika took these for further refinement, modeling, and interpretation. And this is what she found. So this is spike, this is RBD, and this is a monoclonal on the top. The data I'm showing you here is for map 13. She's done this for the other 3 maps, but for the sake of time, I thought that let me just show you map 13 because it seems to look pretty interesting. So the 3D, let me first show you the way it binds so you can see how it works. So please concentrate on the monoclonal being in light green and the RBD being in lilac, and hopefully this movie will play. So this monoclonal seems to be very interesting because unlike the other 3 monoclonals, this actually binds from the back of the RBD. And so it's a unique portion of the RBD that we've not seen. And as this structure will move towards us, you'll be able to see that this monoclonal actually binds in the up position. So this is RBD in the up, this is the RBD in the up, and this is the green color is the monoclonal that is bound to the RBD up. So this looks very promising because it binds to a unique area. It's also a class 3 monoclonal that it's binding to at least 2 RBD ups. In this structure, we feel that it's probably a third RBD is also up. And as you know, the RBD has to be up to bind to ACE2. So we feel that this monoclonal will be, is potent because just the unique way that it binds to the RBD. But if we want to look for therapeutic monoclonals, we did not want to settle with just one monoclonal. One of the main reasons for that is that using a single monoclonal, and if it was used robustly across the population, you might initiate some level of escape mutations within the virus. So ideally, a cocktail of 2 or more monoclonals would be preferable. So Anamika is also working towards doing some level of reconstruction of models of these 4 potent monoclonals to see how they bind. The second thing is if you want to make a cocktail, then you don't want 2 monoclonals competing for the same site on the RBD. So you want to ensure the cocktail contains monoclonals that bind to unique regions, have high affinity for their binding. So I'll show you quickly just to give you a flavor. This is monoclonal 13 and the map O2 that Anamika has done some modeling for. And as you can see that they bind to 2 very unique regions of the RBD, map 13 slightly at the back and map 2 slightly in the front. I'm not sure whether this is clear, but the yellow shades that she has provided here are actually the mutations that are commonly found in some variants of interest. This is the K417N, L452R, and E48Q. So this is where we are in terms of where working towards a cocktail and doing some level of structure determination. Anamika has just taken some time on the New York Structural Genomics Center where she will get higher resolution on a Titan Krios. And so we'll have a little more details about these monoclonals and hopefully further on how when we bring monoclonals together, what their IC50 values look like. Some preliminary data that Mehul has, it looks like when you start combining these 2 monoclonals together, we can bring down the IC50 values by 10-fold. But that data is preliminary. He's repeating the experiments and he's also looking at variants. He's looking at also the Delta variant carefully because that seems to be the most prominent circulating virus as of right now. So with that, I'll quickly summarize what I've shown you, that we made monoclonals by baiting RBD-specific memory B cells from COVID-19 recovered individuals. I've shared data on what these 92 monoclonals look like. 50% of them at least inhibit RBD ACE2 interaction. I shared some data on live neutralizing assays where we found 4 potent monoclonals. The functional characterization is ongoing in terms of whether these monoclonals also neutralize variants, at what IC50 values, and in what combination can we use them. Anamika is working on further structural characterization of these at least the 4 potent ones that we have found right now. So with that, I will stop. And I will acknowledge the people who were involved in the work. Obviously, ICGB admin and the director's office, they are working with the funding agencies to see whether, if we see that these monoclonals are promising, if we can take them from the lab to bedside at some point of time. Sanjeev in the lab led most of the work. He's a welcome DBT early career fellow. Ram did a lot of key experiments here along with Kaustav and Kamal. Amit's lab along with Ritu and Payal are actually doing some amount of X-ray crystallography with looking at RBD and FAB, and hopefully we'll have that data soon. Since we start with single cell sorting, none of this work is possible without a functional good single cell sorter. So a big shout out to ICGB TSCF facility for the single cell sorts during the lockdown when nobody could move out of the quarantine areas. So I really appreciate Aditya for that. Since this is a collaboration with Emory, Rafi and Murli for sure, because my lab is a collaboration with Emory Vaccine Center. Mehul is doing most of all the neutralizing assays. Jens, Carl, and Reddy did a lot of hand holding when we started to make the monoclonals. And Anamika and Eric's structure is what I shared right now. So with that, I'd like to thank you for your time. I really appreciate this opportunity. Quickly to put names and faces, this is Sanjeev, this is Ram, Kaustav, and Kamal is at the back. And this is the entire group at ICGEB who were some way or the other involved in the work. And I'll stop here. If we still have time, and if there are any questions, I'd be happy to take them. Thank you.